Good morning, everyone. My name is Brian Lun. I was a full-time biology teacher in a local secondary school for 4 years, and I taught two full cohorts of the biology class. Some of them get a really good results, like level 5 or above. And although that school would be a band-free local schools, still it would be a very proud result to both of me and their students. For the teaching and learning qualities, I actually have the teacher registration, and I got a distinction in my practical, in a university life practical. Um, for my teaching style, actually, I would describe myself as exam-oriented teaching style. It is because I focus a lot on how students how they answer questions, how the keywords is well explained, how their ideas is clearly illustrated, how the diagrams is drawn. I would say I focus a lot on exam skills. That's why I would say myself would be an exam-oriented teaching style. Of course, I am a proactive and friendly person, which means that I actually focus a lot on my teaching and learning. However, because sorry, however, because I am not liking on creating activities for students, or not creating, like leading some students to the outside activities or some administrative works. Usually, I would love to focus on my teaching and learning, which means that I really wants to join the Canada education team, because in Canada, education comes first. All the other administrative works or other stuffs would have another professionals to handle their professions. Also, it would provide a favorable environment for English learning and teaching. For me, usually because I was in local secondary school for 4 years, and I've been into English, the whole Chinese without any English environment for 4 years, leading me to have a little bit of um, um, disappointment on my English teaching and learning. And I want me myself to keep improving. Then the best way would be sticking with the English teaching and learning environment. So joining kind of would be a good way for me. Of course, joining kind of would means that professional team, we are working together. All of us are coming from top schools, top colleagues, and which means that our knowledge would be the best, and we are having our professions well. So after introducing myself and telling you why I'm wanting to join Kana, so perhaps we will start my lesson. So before starting my lesson, because I am exam oriented person. Usually in my lesson, I will first show students the prior knowledge, they have, uh, what we have discussed before, and what we're going to discuss later, which means that the learning objective, which is both are the linked materials, and then I will simply provide a examination question, like a public one, or the I make up one question for them, so as to allow students to understand their skills are well equipped or not, or their learning objectives are well learned or not. So this would be my approach. Let's do it. Good morning, everyone. So today we are going to discuss on the autosomal gene linkage dihybrid cross, which is one of the Mendelian traits. Last time, the previous lesson, we have discussed about the monohybrid cross, which is one of the Mendelian traits Today, we are going to add another one, which is dihybrid one. Still, we have to stick with the Mendelian traits. Later, like next week, we'll discuss the non-Mendelian traits and the pedigrees later. Okay? So let's begin. So today's lesson flow. Today's we are running out of time. We have still only 15 minutes left, which means that we have to go through very quickly. First, we have to discuss on the prior lineage, which is the dominant recessive allele or the one trait monohybrid one, like previously speaking. And today we're going to learn how to predict the genotype and phenotype of dihybrid using Punnett square. So monohybrid, which means that we are simply discussing one trait. So for dihybrid, we're talking about two traits. However, we're still using Punnett square. So let's keep up. Last time, we are talking about a Guinea pig, like the hairy allele and the hairless allele. So if we still remember what is called homozygous dominant or homozygous recessive. Yes, Savio. Yes, of course. So thank you for your answer. Savio has mentioned a very good point is that when we are having homozygous dominant, which means dominant, which means that this trait will show first, okay? So for homozygous one, which means that both of it would be capital letter, okay? So for recessive one, which means that both would try to hide themselves, right? Hide themselves. So we are trying to write down small letter, like hairless, which means H, okay? Both are H. Homozygous, which means that both of the alleles are the same. Okay, yes, Savio, you're correct. So would maybe Christina, can you try to write down the heterozygous, the genotype of the heterozygous one? Yeah, a capital H and a small letter H. You're correct. Thank you very much. Both of you are doing a great job. So let's check. We are all sticking together and we are doing good. So trying to form up a heterozygous parent and a homozygous recessive parent and trying to do it in a Punnett square, trying to predict the genotype and the phenotype of both of the offsprings. If you still remember, so the top one would be on top of the Punnett square would be the gamete. Which type of gamete will have from the parent? Okay, let's do it together. Okay, we have passed a minute, so let's check together. So when we are having the Punnett square, make sure every offspring consists each of the parents, one of the gametes from each of the parents. So assuming we are having the capital H and the recessive H from each of the parents, then we will form up one capital and one small letter, which means that this kind of genotype would be capital H and small letter H. Okay, for phenotype of this one, because we are showing the dominant first, that's why the genotype would be the dominant the phenotype would be dominant one, which is hairy one. And we can see that according to the Punnett square, and we have the genotype ratio and the phenotype ratio on the right hand corner. So to check if you are doing it correctly. All right, so we'll move on. So from the previous one, we are having the one trait only, the monohybrid one. So what if we add another trait? This time we are having another situation. Assuming cats both have one of the trait would be hairy and hairless, like Guinea pig. Another trait would be sync loving and non-sync loving. Okay, so this cat would love to stick with the sync, which means that sync loving. Another trait would be non-sync loving, which means that they do not like to stick with the sync. Okay, so sync loving would be the dominant one, which means that we are writing it as capital S, and non-sync loving, we'll write it in small letter s, okay? So let's guess. Oh, sorry, I've moved too fast. Okay, when we are writing the genotype of a single cat, which consists of both hairy traits or single loving traits, actually we'll write it into the same column. Let's say for the first one would be hairy trait. Another one would be sync loving traits, and we put it together. Okay, and we put it together. However, we consider them as each of the independent traits. So each trait consists of 2 alleles. Okay, of course. So we can both as one of the homozygous traits, or another one would be heterozygous. Both are fine. Okay, so assuming we are having both parents as heterozygous. Okay, both traits are heterozygous. Let's guess what would be the gamete combinations which you put on top and the left side of the square, which means that both columns consist of 4 types of gametes. Let's guess which 4 types. I'll provide you one. Let's see, because we are having both kinds of traits differently. So both kinds of traits are heterozygous. So for the first one, which means that it would be the hairy one, we will have the capital one, and we will have the dominant one and the recessive one. So we have 2 kinds of different stuff. So assuming the first type of gamete will have the hairy one, the dominant one, another one would be recessive one, okay, the hairless one. So another type would be if it is sync loving. First one would be sync loving, another one would be non-sync loving. So is there any possibilities to have 2 more types of gametes? Okay, let's guess. Okay, so after a minute has passed. So let's check together if you are doing correctly. So actually, because both of the parents are heterozygous, that's why we have the same combinations for each of the column. And perhaps maybe Ronald, yeah. Can you please explain to me what is foil principle means? Yeah, foil principle means that's how you describe and how you write down the combinations of gametes into the Punnett square. So, as you can see, I was trying to describe as capital H, capital S, and then the small letter h and small letter s. So this would not be correct. This would not be correct. We must follow the foil principle, which means that first outside, inside, last. Okay, this would be the correct one. So try to avoid the mistakes that I have just shown to you. All right. So assuming we are having the Punnett square that I've just shown you, what would be the phenotype ratio according to the Punnett square? Let's try. Okay, I'll give you simply like 2 minutes trying to come up with your answer, trying to discuss with your friends, with your classmates. If you have any question, just raise your hand and please let me know. Okay. All right. So after 2 minutes has passed. Let's check if you are doing it correctly. Perhaps maybe Christina, can you please come outside and try to write down the answers that you have come up with? All right. Thank you, Christina. So Christina's answers is actually pretty much the same as mine. So Christina's answers actually come up with the phenotype ratio of 9331. What is meant by 9331? Actually, so for 9, which means that both of the traits are showing dominant 1. Why is showing dominant 1? Because each of the offspring consists of each of the traits with at least one of the dominant allele. So we have dominant. So all of this have at least one dominance. So according to this Punnett square, all of the 16 choices, all of the 16 probabilities, we have 9 of it consists of both dominance, at least one dominant allele. So we're showing the dominance traits. That's why it is 9. So 3, 3, 1, actually pretty much the same. So for 3 is dominant and recessive, and so on and so forth. So come up with the Punnett square, we have 9, 3, 3, 1 with the ratio of 9, 3, 3, 1, which means that it is dominant, recessive, dominant, and recessive. So this would be the phenotype ratio accordingly. So if we are going to have, because we are running out of time, so perhaps we will not go deep with the principle behind first. We'll try to complete a examination question. Let's see if you can trying to come up with the Punnett square correctly this time. So let's try to take a look with this question. So in maize, grain color and grain texture. So we can see that grain color is a kind of trait. Another one would be grain texture. So these two are controlled by 2 different genes. To determine whether the inheritance of grain color and grain texture follow Mendel's second law, which means the law of independent assortment, okay, which is this principle behind. A scientist performed the following process. So we can see we have both parents, okay? We don't know if it is homozygous or heterozygous. We don't know, okay? And they will complete the crossing, have the fertilization, and give a cohort of offspring with the F1 generations. So it would be all yellow and starchy grains. So according to what we have learned previously, so exclude all the F2 generation first. When we have both parents and showing the F1 generations with the same type of, color will be green, okay? All with one kind of trait, the same kind of trait, which means that they should be homozygous. So we know that both are heterozygous, both are homozygous. So yellow starchy, which means that assuming we are using capital letter for yellow and green color and T for texture. So it would be another one would be YYTT. Okay, so when these two parents crossing together, your F1 generations would be, so that's why we can answer with the character which one would be dominant, which is yellow and starchy. So we simply pass that question first. Let's finish this question together. So draw a Punnett square to show the cross of F1 plant using YY and TT. Okay, so color and texture would have two different types of symbols that represents. First one, we have to come up with the parent first, right? So both parents okay, so both parents are F1 plants. So F1 plants, we are having both heterozygous one. So actually, simply like the same as previously we have mentioned before. Both are heterozygous. So let's write down the gametes first. Assuming we have the gametes. Okay, sorry. We have 4 types of gametes if we are remembering correctly. Okay, 4 types of gametes. So first one, because we are having YYTT, right? So using the foil concept, first, so YT outside, YT, and then inside, and then the last. Okay, so both are pretty much the same. So complete the Punnett square together. So trying to complete for me. So this would be your homework. And this would be the end of my lesson. So we will have the learning objective checklist after we have finished the lesson. So after you finish the lesson, actually I will trying to go through with them the answers if we are doing it correctly and try to show them the learning objective checklist to allow them to check if they are completely satisfied their goals of today's lesson. Okay, so this is the end of my lesson. Thank you.